about, I think this is a different one, so not an issue, but Android. Meromycin, it is made up of two polypeptide chains. It is made up of two polypeptide. Okay, which fish takes parental care in mouth about baby fish other than hippocampus? Some random person asked me this question. Beta, I don't have any idea about that. Sir, फिर से explain करो. Sure, I will do definitely. I think now it is all right, I think. Okay, so one meromycin has got two polypeptide chains. One I've drawn the polypeptide chains with different color, one with white, one with green, so that all of you can appreciate the two polypeptide chains, isn't it? Right. So I said it forms the tail part, neck part, head part. Neck part and head part complete, we can call it as cross arm. Cross arm, this is the important point. Cross arm and the tails, this is the tail part. The tails, okay, these are the tail part, tails, and tails of them, they are helically twisted. Tails are helically twisted, and then the neck and then the head. Now, one of the binding site in the head is for ATP, that is ATP binding site, okay, ATP binding site, and one more binding site, okay, so there is one more binding site, and this is that one more binding site, and here this is for actin binding site, actin binding site, and this is ATP binding site, actin binding site. So each of the head has got two sides, one is for actin, one is for ATP. Now whenever ATP binds here, it can catalyze the hydrolysis of ATP and ADP and inorganic phosphate, and energy can be released. So bachcho, can I call it as enzyme? Now tell me, who will tell me, can I call this as enzyme? Obviously, can I call it, is it an enzyme? Ha beta, it is enzymatic action, isn't it? Tell me, can we call it as enzyme? The head part, yes, it is an enzyme. It is ATPase. Very good. The enzyme is ATPase. Very, very good. It is an enzyme. It is an enzyme action, and the enzyme is ATPase. What the ATPase does? Same thing. ATPase, any ATPase in the body does the same thing. Hydrolyze the ATP into ADP and inorganic phosphate. The high energy bond, when it is broken, large amount of energy is released. Yes, it is ATPase. I hope this point is clear. So if they ask you which part has ATPase action, very clearly you can say the head part has ATPase action. Okay, then they try to trap you. Okay, tell us if the head is not given and they ask you that which part, is it a cross arm or tail? Very clearly cross arm. Now today we know the structure and directly we have drawn the structure. Just imagine one day when they did not have the exact structure with them. So at that time also they found that it has got ATPase activity. So they wanted to locate which part is that. They use an enzyme called trypsin and try to break the parts, okay? So they can just break it into two parts. So they can break it into two parts like this, and so they call this part as HMM, heavy myromyosin, and this part is LMM, light myromyosin. Heavy myromyosin, light myromyosin, and heavy myromyosin, this is having the head and the neck region. Today we know that, and the light myromyosin is the tail region. Now if they ask you which part has ATPase activity, obviously heavy myromyosin has ATPase activity. Neither the tails and nor the LMM, they don't have the ATPase. So many questions can be given about ATPase, and which ion ATPase use in our body. ATPase uses magnesium as cofactor. Generally magnesium, animal body does not use magnesium, but here magnesium is used as a cofactor. Is the point clear to everyone? Clear ho gaya? Haan, Mg2 plus, very good. So bachcho, this is all about the myromyosin. 300 of these myromyosin. Now you can see them as two chains, okay? What I'll do, I'll just zoom out. I'll just zoom it out. I'll just zoom it out, okay? I'll zoom it out, zoom out. And ultimately I can see this as, if I want to draw it, it comes something like that. Isn't it? It comes something like that. Or maybe this is helical. So actually these two chains, I mean, when I zoom it out, it look like that. So just imagine that how the thick filament is formed. Thick filament, these are the cross arms, okay? How the thick filaments is formed. 300 of these molecules, bachcho, 300 of these molecules are forming the thick filament, and their tail parts are interwoven, and both the sides they are forming the cross arm. And you can understand why we call them cross arm, because these are going out of the main axis. They are going out of the main axis. Okay, bachcho? So this is a thick myofilament. This is a thick filament. Thick filament. I hope this point is clear to you. Thick filament, and these are the cross arms. Cross arms, okay? And at this point, there is a membrane. There is a fibrous membrane. A fibrous membrane is attached here. A fibrous membrane is attached here. A fibrous membrane. Which membrane? Fibrous. A fibrous membrane is attached here. Anyone know the name of this membrane? We call it as a line. What line we use it? It's a fibrous in nature. A fibrous membrane. This is very important. It is fibrous membrane. Fibrous membrane. What do you call it, bachcho? Fibrous membrane. I know that meanwhile, membrane. I know that by the time I'll go there. Yes, this is called M line. M line, good. This is called M line. So thick myofilaments are attached to the M line, good. And thin filaments are attached to the Z line. Thin filaments are attached to the Z line. Thick filaments are attached to the M line, and thin filaments are attached to the Z line. M line is fibrous in nature, Z line is elastic in nature. M line fibrous, Z line elastic. M line fibrous. So one is M line, it's a fibrous membrane, fibrous membrane, and Z line. Z line is an elastic membrane. It is an elastic membrane. Elastic membrane. Fibrous membrane. M line attached to thick, or all the thick filaments. Thick filaments are attached to the M line. To the Z line, thin filaments are attached. Thin filaments are attached to the Z line. Please remember this summary. Please remember this summary. Fibrous membrane, elastic membrane. Fibrous membrane, elastic membrane. M line, Z line. M line is thick filament. Thick filaments are attached to the M line, and thin filaments are attached to the Z line. And therefore, when the thin filament slide over the thick, the two Z line, they come closer. So, bachcho, this is a structure. I think I told you about the thin filaments, na? Thin filaments, I can remember thin filaments, tell me. बताओ, thin filaments हो गया? ठीक है. Got it. Very good. Very good, beta. And thick filaments, nearly 300 of them. Nearly 300 meromyosin. This is one meromyosin. Meromyosin means the mere. Mere means unit. It is a unit of myosin. Meromyosin means unit of myosin. I hope it is clear. Okay. So if I make it bigger, if I enlarge it, you can see the meromyosin. I don't think so. Anyone, the type of students you are, you will have any problem in this understanding. You all can answer That's what my, yes, it is a polymer. Very good, Anjali. And now let me talk about sliding filament theory. Very interesting. It's called sliding filament theory. Sliding filament theory. Okay. Now, how the contraction occurs, we understand it by sliding filament theory. Sliding filament theory. Okay. Now, whatever is in the diagram is minimum, like I have to make. Without that is not possible. That I will make, or else I will use the diagram of the books only. Right. Now, sliding filament theory. The sliding filament theory says that because the myosin head, the moment they make a contact with the myosin heads, the moment they make the contact with the actin filament, now actin filament has the active site. So the actin binding site, when it attaches to the actin binding site of myosin head, attached to the actin filament, then after that the cross arm take a power stroke. It means that the cross arm bend. Now, in which direction they bend? All of them bend to the middle of the A band. All of them bend to the middle of the A band, so that the thin filaments start sliding towards the middle of the A band. The thin filaments, this side also slide towards the middle of the A band. Is the point clear to you? Okay, Malli, I'll tell you this diagram of our NCT diagram only, if possible, or else I'll use the diagram of the book to show you how this sliding filament, and then we will go to the minimum thing that I said, that diagram. I will use the diagram to explain you. Okay, so just now I will use here image pen drive. I think I used this black wala pen drive, no? Last time. Biotech. इसमें muscular movement, muscle is not there. It's not there. This is good one, contracted sarcomere and relaxed sarcomere. So very, I mean, it's colorful also, and it is good. I think this will make you understand better, something very colored and something very very attractive. You can now locate the Z line, M line, locate the different bands are there. So there is a band, which is a dark band, and I band, which is a light band. Okay, sarcomere, we are now doing the sarcomere, beta. Different part of the sarcomere, you know that sarcomere is a unit of muscle contraction. Sarcomere is a unit of muscle contraction. You can see this part. This is the A band because it is a dark band. Dark is anisotropic band. Anisotropic. So to make it easy, we can call it as A band. A band. So from here to here is the A band. A means anisotropic band. Now from this point to the another one, this point, this is the light band or I band. This is the I band. I'll label it here from this point to this point. This is the light band, also called I band. To remember, the vowel I comes in the light and A comes in dark. So a dark is A band, an isotropic band, and light is I band, isotropic band. Okay, so these are the names of, and similarly, beta, what is this called? This one is, because alternate light, dark, and this is also light band, I band. Light is I, dark is A. Okay, so one sarcomere is from Z line to another Z line. This is one sarcomere. One sarcomere means it is a unit of muscle contraction. Sarcomere, one sarcomere is from Z line to Z line. So, what are the different parts of a sarcomere? Video nahi dikh raha. The portion black and white, two lines. Okay, ठीक है. Okay, बच्चों, very good. Now please understand. One sarcomere from one Z line to another Z line. ठीक है? तो Z line, I band, A band, I band, Z line. Z, I, A, I, Z. And since the Z line is bisecting the light band, actual light band is from here to here, and you find the Z line going in between. It is bisecting, dividing into two. The Z line is dividing into two. So dear children, please understand that this is the half of the I band. Then Z line, half I, full A, half I, Z line. Z, I by 2, A, I by 2, Z. Okay, sarcomere. Z, then I by 2, then A full. Again I by 2, and this is the Z line. This is one sarcomere. When we understand the contraction of one sarcomere, it means that the whole muscle contracts. So this is the unit of muscle contraction. But if you ask me the structural unit, the muscle cell or the structural unit of the muscles as our textbook has given. Okay, so Z line is in the middle of the I band. Very, very good. Beta, this is one thing. Okay, now when a sarcomere contracts, what happens? The two Z lines come closer. So what are reduced? Let me explain that. So you will one more thing. Last point. A very important point is that in the A band there is a small part. A part in the A band where only thick filaments are there. Means here. A band में कुछ ऐसा part है, this part from here to here, where only thick filaments are there, no thin filaments, no, or there is no overlapping. This region is called the H zone. This is called H zone. This region is called H zone. H zone is the part of the A band having only one type of filament, which is the thick filament. Okay, and in the middle of the H zone there is an M line. Okay, so we can make n number of statements from here and we can understand. Okay, so only thick filament, part of the A band, only thick filament is known as H zone. Okay, is the point clear to you? So when a muscle contracts, actually the sarcomere shorten. So when the sarcomere shorten, you can see down, this is a contracted sarcomere. When the sarcomere contracts, you will find that this I band is reduced, right? So I band is reduced. Let me write here, I band is reduced. Then you will find that H zone is also reduced. H zone is also reduced. I band reduced, H zone reduced. Okay, I band reduced, H zone reduced. In fact, H zone afterward disappear also. I band reduce, H zone reduce. A band remains unchanged. A band is this only. A band remains unchanged. A band remains unchanged. And why the I band reduce? Because the thin filament slide over the thick filament. Why the I band reduce? Because these thin filaments, they slide over the thick filament. Thin slide over the thick. Thin slide over the thick. Reason being, this cross arm will pull the thin and pull them towards the middle of the A band. Okay, so because this cross arm, a cross arm, when they make a contact, they make a bridge between thin and thick, and they pull them in the power stroke. They pull them towards the middle of the A band. Is this point clear to you? Even if 4-5 members write, I will understand that this point is clear to you. During a muscle contraction, sarcomere length reduced, I band reduced, H zone reduced, A band remain unchanged. Okay, okay, very good. हो गए 5. ठीक है, clear. Now tell me one question. Very good. Tell me one question, that when a muscle contracts, what happens to the length of individual myofilament? Question. Thin filament reduced, thick filament reduced. Option 3 is they remain unchanged. The length of myofilament remains unchanged. Okay, length of individual myofilaments remain unchanged. Yes, correct answer is 3. Length of individual myofilaments. बेटा चूड़ू, this myofilament thin, ये अभी भी उतना ही है. It's not shortening. The thick myofilament, it is not shortening. So length of individual myofilament remain unchanged. Answer is 3. Can I say that the A band and the length of individual myofilament remain unchanged? Can I say that A band and length of individual, ये मैंने पहले बता दिया, already told. Okay. सार्कोमियर हो गया ना, सार्कोमियर. ठीक है. I'll tell you the sliding filament theory. Sliding filament theory, shall I tell you? Sliding filament. ठीक है, स्लाइडिंग फिलामेंट करें. वेरी गुड, स्लाइडिंग फिलामेंट वी आर डूइंग. अल्लम चटनी सांबर बज्जी हो तो गया. बच्चे कह रहे हैं वी शुड एक्चुअली स्टार्ट स्लाइडिंग फिलामेंट थियरी. शुड स्टार्ट ही. अच्छा, बाय मिस्टेक we started all that. Anyways, bachcho, just listen here. Sliding filament theory, sliding filament. Hey my God, I understood that. I only said to you that remind me about the LMM, HMM, and I thought that the thick myofilament completely I have to show, because in the beginning someone write. Anyways, it is a confusion. Don't worry. Now what happens that this cross arm, they can form a bridge between thick and thin. Now when that happens, please understand that as long as these are covered, it can never happen. As long as these are covered, it can never happen. And in the resting muscle, they are always covered. In the resting muscle, they are always covered. The active sites of the actin are always covered. But when the muscle is depolarized, when the muscle is depolarized, when the muscle is depolarized, then calcium will come out of their storehouse. Calcium storehouse are sarcoplasmic reticulum. Calcium comes out from their storehouse, and they will bind to the troponin, and then the inhibitory effect of troponin is gone. Inhibitory effect is gone, and at that time the tropomyosin will uncover the active sites. Now you can see that. You can see here that tropomyosin have covered you can see here tropomyosin have covered the active sites like that. You can clearly see they have covered the active sites, and in this diagram you can clearly see that they have uncovered it. Now why that happened? Because the calcium ion bind to the one of the subunit of troponin. The calcium ion bind to one of the subunit of troponin. And also you can clearly see that what is happening here is the contact. This cross arm is joining or making a contact with the active site. What are these? These are the active sites on F-actin, and actin binding site will join the active site. You can see that the actin binding site, the actin binding site of here, this is actin binding site of the myosin head is actually making a contact with the thin filament. Now this is a bridge formation. The bridge is by the cross arm. So the bridge can be called as cross bridge. The bridge is by the cross arm, and we can call that bridge as cross bridge. Okay, bacho. So I think you have understood here. Now what happens is when the bridge is formed, then this folds. When the bridge is formed, then this folds. This is fold. When the bridge is formed, it folds. When the bridge is formed, it folds. All of them, they fold or they bend towards the middle of the A band, towards the middle of the A band. So the direction is this direction. They all fold towards the middle of the A band. Because of this, this thin filament starts sliding. The thin filament starts sliding over the thick filament. But right now it may look it may look like thick, but actually it is a thin filament, and this is a thick filament. The thin slide over the thick. Please understand that there are two options: thin slide over the thick, and option number 2 is thick slide over the thin. So please understand that thick remains there only, and the thin is sliding over the thick. Therefore, we call it a sliding filament theory. But then question is how the membrane become depolarized. The muscle membrane, now you have to tell, sir. Okay, muscle membrane is sarcolemma, and how it become depolarized. First of all, during resting, it is polarized. Okay, and you know that when a nerve impulse or action potential comes, then it becomes depolarized. Now depolarized means that now the charges become opposite. I'll just quickly, because I cannot remain without making somewhat diagram on board. So that is, I have to show a little bit in sliding filament theory. So bacho, this is a nerve impulse. First of all, the motor neuron, nerve ending, motor neuron, nerve ending, okay? And we have a muscle fiber. This is a muscle fiber, and a muscle fiber at rest is a polarized one. At rest, the muscle fiber is polarized. So I am showing the sarcolemma. This is sarcolemma, and this is polarized at rest. Means that outside it is positive and inside it is negative. When we say polarized, it is positive, it is negative. And you know that there are some vesicles here, means membrane bubbles. Inside this membrane bubble, acetylcholine, which is a neurotransmitter present inside these bubbles. Acetylcholine, which is a neurotransmitter, is present. And my dear children, you will find that the acetylcholine receptors are lying on the membrane of the muscle. Acetylcholine receptors. So these green color thing that you see, these are the acetylcholine receptor. What is acetylcholine? Acetylcholine is a neurotransmitter. Where it is present? It is present at the axon terminal. It is present at the axon terminal. From where this axon terminal comes? So try to understand that. So motor nerves, you know that motor nerves are attached to the muscle. So this is the fiber. Okay, bacho, this is a muscle fiber, okay, and this is a motor neuron. This is a motor neuron, and this is a muscle fiber. Motor neuron, muscle fiber. This complete is known as one motor unit. This is called one motor unit. Sometimes there can be more than one muscle fibers. Sometimes there can be more than one muscle fiber. So in one motor unit, what is one? Okay, I tell you, there is a motor neuron and n number of muscle fibers. N number of muscle fibers. N number of muscle fibers. N can be one or more than one. N can be one or more than one. You have to tell me what is one in one motor unit. Come on. अब कहते हैं ना एक मोटर यूनिट है तो व्हाट इज वन इन दैट What is always one in one motor unit? Narayan Sai has given, I think, misprint हुआ बेटा, motor neuron plus n number of muscle fiber. Yes, motor neuron. Very good. So what is one here is one motor neuron. Motor neuron. And in motor, this is a motor neuron, efferent, and this can be n number. N can be one or more than one. N can be one or more than one. This is one motor unit. Important point. Okay, beta, there are certain functions. For example, like our fingers. In fingers, very precise actions are happening. So generally, the motor unit comprises of very few number of muscle fibers. One motor neuron, few number of muscle fibers. But there are certain places, for example, the back muscle. Now, back action is not a precise action. It is a contraction so that we can stand. Our back can remain straight. So there is this contraction, and when we lie down, it relaxes. So it is a very precise, no, it's not a very precise action. It's a coarse action. That means all have to work in the same way. Now here is very precise. My mind is controlling, and the way I move my fingers, I can make the different stroke, which can give different meaning. But that we are not doing with the back muscles. So in the back muscle, one motor neuron has many muscle fibers. And in these muscles, one motor neuron has few number of muscle fibers. Right. So one motor unit, I hope you understand motor unit. One motor unit में one क्या है? One is the one motor neuron, efferent neuron. And this is the junction of the efferent neuron. I have shown you. This is a junction here. This is a junction here. This junction is known as myoneural junction or neuromuscular junction. This junction can be called neuromuscular junction. I'll write here neuromuscular junction. Neuromuscular junction. Beta, NCERT textbook writes, it can also be called, I'll write like this. It can also be called as motor and plate. Motor and plate. Now these terms, please bachcho, we should not lose marks in such terminology. No one here, we should not lose marks in so simple terminology. Motor neuron, if we know that, motor unit also, and motor and plate. Neuromuscular junction, motor and plate. Okay, bachcho? Now some people are asking about this, that why the muscles, one motor neuron is controlling less number of muscle, which means that in this area, so many motor neurons are coming. In the back area, few motor neurons are sufficient to make them contract, but in this area, many motor neurons are there, isn't it? So think where the action can be well coordinated. It's simple. So it's not a rocket science. यहाँ पे कोई coordination तो है नहीं, contract हो गया, वही खड़े हो गए. You understand the point? I hope it's clear to everyone. Wonderful. Beta, let me just focus you here. What happens when the nerve impulse come at this end? Now, this is important to note. When the nerve impulse come at this end, so these vesicles, they will move towards the periphery. I have made a wrong thing here because I should use the same color to show the vesicle. Same color. Why? Because all membranes are same. We call them biomembranes. All membranes are unit membranes. They have the same structure, right? So lipid bilayer, phospholipid bilayer. So and they are fluid in nature. So when this membrane touches this membrane, both of them fuse like two small bubbles fuse to form a big bubble. So bachcho, when nerve impulse come here, how should I draw it? I'll show you. When the nerve impulse come here, I'll show you how this looks like. Okay, so when the nerve impulse come here, this will look like this. The same one is I mean, different stages I have shown. I have shown some coming closer. I have shown that some of them have almost fused completely. Some are almost fusing. And these are the vesicles containing the neurotransmitter. The neurotransmitter here, neurotransmitter, and here the neurotransmitter is here. Here it is coming, and this is how. And it is now released in the synaptic cleft. In this junction, the neurotransmitter is released. Now you know that its receptors are lying on the membrane of the muscle fiber. Its receptors are lying on the membrane of the muscle fibers. So these receptors are precise receptors for acetylcholine, and I can show that this acetylcholine binds to here. Acetylcholine binds here to the acetylcholine receptor. The acetylcholine binds to the acetylcholine receptors, and you know what happens? The permeability for sodium ion increases. Sodium permeability increases. Sodium permeability increases. Now, bachcho, when sodium, and why this happen? Because the acetylcholine bind to its receptor. Then permeability for sodium increases. A massive influx of sodium ion, and sodium ion carries a plus charge. So the muscle cell from inside become positively charged, and this stage is called depolarized. Mundu polarized, now depolarized. Why? Sodium permeability has increased, depolarized. Now, when the muscle membrane become depolarized, the calcium doors open up. The calcium doors open up. Calcium is stored in it's like packed inside the sarcoplasmic reticulum. It is packed. Now the doors open up. When the calcium doors open up, calcium ions start coming out because of very high concentration inside the sarcoplasmic reticulum. So calcium diffuse out of sarcoplasmic reticulum. They were not coming because the doors, the channels were closed. But when the depolarization occurs, channels open up, and calcium comes out of sarcoplasmic reticulum.
Absolutely.
Coming now, promise. Tulsi, my promise. My promise. Tulsi is saying, sir, please this time you can teach till the end of the chapter. Okay, promise. Why it causes stiffening of the muscle, rigor mortis? First you understand the contraction. So what happens, the nerve impulse comes, the nerve impulse makes the release of the neurotransmitter in this cleft, in this gap. Neurotransmitter bind to the receptor. These are neurotransmitter receptor. The neurotransmitter is acetylcholine. These are the receptor for acetylcholine. And there is an increase in the sodium permeability. Sodium ion, influx of sodium ion occurs, and the membrane now become positively charged from inside, and comparatively outside is a negative charge, and now inside become positively charged. You know that this stage can be called as depolarized. We call it as depolarized. Depolarized, okay? Earlier was polarized, and now we can call it as depolarized. You can clearly see this. You can clearly, bachcho, see this, okay? It is depolarized, so inside become positive, outside become negative, and then what happens now? What happens? Because of this depolarization, the calcium channels open up, calcium comes out, calcium binds to the troponin. Calcium binds to the troponin, and the inhibitory effect of troponin is lost. Calcium binds to the troponin, and you can clearly see the inhibitory effect of troponin is gone, and that's how the tropomyosin have uncovered. They have uncovered the active sites. The tropomyosin has uncovered the active sites. I hope you understand how the calcium becomes available, because of the depolarization. Calcium binds to one of the subunits of troponin, and as long as calcium is there, please understand this last line very, very carefully. But as long as calcium is there, please understand this line very carefully. As long as calcium is there, this will remain uncovered, exposed. जब तक calcium रहेगा, it will remain exposed. Active site will remain exposed. Clear? But what happens that the depolarization is followed by repolarization, and this is automatic. The depolarization is followed by repolarization, okay? And when the repolarization occurs, there are some active pumps. They take all the calcium from the sarcoplasm, and then they put them inside the sarcoplasmic reticulum. Active pump, they take all the, and sarcoplasmic reticulum is too much all around the myofibril. They take all the calcium, and they lock them inside sarcoplasmic reticulum. This occurs actively, okay? And what will happen now when calcium is gone, bachcho, because of repolarization, calcium gone inside, and the inhibitory effect of troponin again comes. Now the troponin and tropomyosin have covered them again, and because they have covered them again, because calcium gone, they have covered them again. What will happen? The myosin head will detach now. Myosin head will detach, matlab toot jayega, and you will find that these thin filaments start returning back. The thin filaments start returning back, and they come to the normal position before, relaxed position. They will return back, the thin filaments will return back when myosin head will detach. You can see that myosin head are no more. And why this no more? Because the active sites are covered by troponin and tropomyosin. The main commanding protein is troponin, and tropomyosin is the one that is actually covering all the active sites. Okay, beta. So this is how the muscle contracts and relaxes. Now I'll tell you the four steps of this cycle. Four steps: when ATP hydrolysis occurs, cross bridge formed, power stroke, and then again breaking of the cross bridge. And there is the reason why the muscle remain contract after the death. Okay, bacho. So now I'll tell you that at one myosin head, let me make a thick filament. In this thick filament, this is one cross arm, one cross arm, okay? And each cross arm has how many active sites? One for ATP, one for actin. One for ATP, one for actin. And beta, this is the thin filament. This is the thin filament, and thin filament has active sites. So I'll tell you where are these active sites on the thin filament. There are active sites. I'll tell you A, and I'll tell you this is the B. Okay, I'll tell you this is C. These are the active sites. Active sites on thin filament. This is thick filament. This is thick filament. Everyone please focus. I know you are tired, don't worry. The more tired you are, the more chances of your selection. The more pain, more gain. I always believe in this fundamental. The more pain, nothing is ever achieved in a comfort zone. Nothing is ever achieved in a comfortable, cozy zone. You have to become uncomfortable. Okay, so take 3 deep breaths and concentrate. Now what happens that already, because you can see the active sites are exposed, that means depolarized already, it is depolarized, and the calcium, it is depolarized and calcium has bound to the troponin, and these are exposed active sites. This has 2 binding sites, one for ATP and one for actin. When the ATP binds here, you know that this is an ATPase. This is an ATPase. ATP binds here, and then it is turned into ADP and inorganic phosphate plus energy. So here I can write here that this step I call it as ATP hydrolysis. ATP hydrolysis. ATP hydrolysis. ATP hydrolysis. Now this is followed by, because energy is available now, and the myosin head will form cross arm. So I'll show you. This is the thin filament, and I've shown the white color, so I'll use the white color. And the myosin head has attached to the active site, and they have attached to the active site thick filament, thin, thick, and active sites. Active sites A, B, and C, A, B, and C, and you have ADP. ADP and inorganic phosphate is here. So I'll make it ADP. ATP has hydrolyzed into ADP. ATP, this was ATP, and now it is hydrolyzed to ADP. This has released energy, and the cross arm, so the actin binding site of the cross arm is attached to the active site, and this we can call it as cross bridge formation. You can see cross bridge, cross BRIDGE, cross bridge formation. cross bridge formation. Step number 1 was ATP hydrolysis. Step number 2 is cross bridge formation. After that, a very interesting thing happens. And you know what happens? So power stroke takes place. What happens? A power stroke. And what happens in the power stroke? The myosin head, or we can say the whole cross arm. The cross arm will bend towards one side. The cross arm will bend towards one side. This side is always towards the middle of the A band. It is always towards the middle of the A band. Now this is interesting, and I'll show you. This is the A, B, C now, ABC, the active sites on the actin, thin filament. There is a power stroke. You can see that this has gone towards the middle of the A band, and very interestingly, you will find that the ATP binding site is again free because ADP, an inorganic phosphate, has been released. ADP, an inorganic phosphate, come out, it will detach from the active site. Okay, but please focus here. ADP plus inorganic phosphate, ADP, these are ADP and inorganic phosphate, and now the ATP binding site is again vacant. ATP binding site is again vacant. This is a cross bridge formation. What you have seen, that this has really bent, and therefore the thin slide over the thick. Thin slide over the thick. This is a step of sliding. The step of sliding due to the power stroke. This is a step of sliding. I'll write here sliding. Okay, sliding. The step of sliding. Sliding because of the power stroke. Power stroke. ADP, and two things are happening. One is power stroke, ADP and inorganic phosphate, they jump out of the active site. Active site again become free, and it is ready to bind with ATP again. Now in the next stage, you have to really understand what has happened actually. The thin filament has slide over the thick, and this is the thick filament. The cross bridge has broken. The cross bridge has broken. The cross bridge has broken. Thick filament. This is a new ATP has bind here. Okay, so new ATP molecule. I'll write here ATP has come, new ATP molecule, and this ATP molecule has bound to its active site. So this is a ATP, this is ATP, and this has broken, and I'll tell you that this is A, this is B, and now it is under C, and this one is further D. Clear? Is this point clear to you, everyone? This is ATP, so I'll change the color. ATP is bound. Remember that ATP is not consumed here. ATP just bound, and this binding has changed the structure of ATP. Please understand that these type of structural changes happen when the substrate bind to the active site. Now this is known as allosteric change or allosteric modulation. Please understand that hydrolysis has occurred here, but here only ATP bound, and there is a breaking of the cross bridge. What has happened here? There is a breaking of cross bridge. Breaking of cross bridge. Okay, so here is ATP hydrolysis. Step number 1, ATP hydrolysis, cross bridge formation, sliding and breaking of the cross bridge. Okay, this cycle is repeating again and again. Till the calcium ions are uncovering the active site. till the calcium is available. Okay, when the calcium go back, when the calcium has gone back to the sarcoplasmic reticulum, then this cycle stop. This cycle keep on occurring till what we find, the thin filament slide over the thick filament. But remember, depolarization is soon followed by repolarization, and after that what will happen, actively the calcium ions, they are pumped inside sarcoplasmic reticulum. Now calcium is not available, so masking resume. Calcium not available, masking resume. Masking resume, right? Resume matlab restart. Masking matlab covering. Masking of the active site on actin resume, right? And you can see that because of the sliding, you can see that this is moving here. And when again it will form this, it will form the cross bridge again. Remember that this won't be B again, because this has broken and come back to the normal position. The cross bridge broke and the cross arm come back in the normal position, and this time this will not be making with the B, this is making with the C. Okay, this time is making with the C. Now this is to be understood, this is B, C and D. So this will make a cross bridge with the C, right? And then it will take a power stroke. Now the C will be here, C will come here, and now the D is available. Okay, so this is C, this is D, and this one is B. Okay, now breaking of the cross bridge, it will again become free, and it will be now, this time it will be under D. So this is causing the thin to move in one direction. So answer the question is which direction, and you know the answer is towards the middle of the A band. Which direction? Towards the middle of the A band. Now these 4 steps, live animation, these 4 steps, help me, sure, pakka Tulsi, I'll open this one, and if Navi ear, this is an animation here. Okay, now please understand that these 4 steps, ATP hydrolysis, cross bridge formation, sliding and breaking of cross bridge. So ATP hydrolysis, cross bridge formation, sliding and breaking of cross bridge. ATP hydrolysis 12 o'clock, then at 3 o'clock, what do you find? Cross bridge. 6 o'clock, sliding. 9 o'clock, breaking of cross bridge. So this cycle keep on occurring. ATP hydrolysis A, then cross bridge formation C, and sliding is S, and breaking of cross bridge is B. A, C, S, B. A, C, S, B. Allam, chutney, sambar, bhaji. Allam, chutney, sambar, bhaji. Allam, ATP hydrolysis. Chutney, your anti-clock, my clockwise. You are seeing me, so my is clockwise only, right? Allam, chutney matlab C, cross bridge, sambar, S, sliding, and bhaji, breaking of the cross bridge. So these 4 steps, allam, chutney, sambar, bhaji. Clockwise, allam, chutney, sambar, bhaji. Allam, chutney, sambar, bhaji. Allam, chutney, sambar, bhaji. Now these 4 you need to understand, and they may ask you to label it, and please, this step is to be understood very, very clearly. I'm saying this step, that during the power stroke, the ADP is released out. during the power stroke, the ADP now it comes out and making the active site vacant again. And because the active site is vacant again, new ATP molecule can bind. And this ATP molecule, just the binding of the ATP is sufficient to break the cross bridge. After death, what happens that these ATPs are utilized. No more ATPs are left. After death, no more ATPs are left. Is the point clear to you? Okay, and because the sarcoplasmic reticulum, they no more keep the calcium inside. ATP required to keep the calcium inside. ATP required to keep the calcium inside. It no more keep the calcium inside, and calcium comes out of sarcoplasmic reticulum. Calcium comes out. This calcium bind to the troponin, and troponin, after calcium binding, will uncover the active site. After getting uncovered, the myosin head, that much of energy, because they already have the ATP bound to it. Okay, so myosin head will combine to the active site on actin, making a cross bridge, and myosin head, after making a cross bridge, take a power stroke, and now after the power stroke it tried to break but unable to break. This step doesn't take place. So muscle undergo contraction. After the death, muscle undergo contraction. This contraction of muscle after the death is known as rigor mortis. This contraction of muscle after the death is known as rigor. It's called rigor mortis. Rigor mortis. I have given you a very simplified version of rigor mortis. However, some more complexities are there in that, but yeah, more or less, this is like this only. Because ATPs are no more left, the breaking of the cross bridge cannot take place, no doubt. After that, when the decomposition starts, then so everything is broken. After that, when 12 hours later, when the decomposition start, then they start loosening. Okay, first time the rigor mortis occur in the heart muscle, but you cannot see that. So what you can see first time? You can see the eyelids. So the eyelids, okay, in the eyelids, rigor mortis appear for the first time. On the eyelids, rigor mortis appear for the first, like you can only see 2 to 3 hours. So if only eyelids are involved, that means the death has happened just 2 to 3 hours earlier. If the muscle is somehow injured, the same cycle continues or not. Why not? Cycle will remain the same. Only thing happens that you will be having so much of pain, so automatically the impulse for contraction will be withdrawn. If the muscle is somehow injured, I have answered that, okay, and allam chutney, sambar, bhaji. Okay, bachcho. So this is all about the muscle contraction. And I also told you how the relaxation takes place. Relaxation takes place by calcium being again pumped inside. We have to start with the skeletal system. And I'll give you certain points on the skeletal system with a guarantee that nothing extra will come out of that, with a guarantee, because I know what can be asked in skeletal system. Because after a thin line, there is a vast ocean, and vast ocean is only for the first year. Vast ocean. Ha beta, this is, I think, Sanam Srisha, this is my pain also. So much of important, almost half of the 720 marks are in biology. 2 periods are not sufficient. I knew that. But what is happening, we were unable. I think now it will be easy because the senior syllabus is over. Today the senior syllabus is over. I was taking one gold batch. So revision will come on alternate days. I think now we can give you time. And last time what happened, on Sunday the test was there. And what the our people said, ki sir, don't take a class on Sunday, because first of all you have to call the people to start that. Well, that is not a big deal. Okay, the big deal is many parents don't find it good that today is the test, and so sir, please don't take a class. And that's the reason last time I just thought that most of you can do that. Very basic things are there, most of you can do that. Extra class, I think Sunday go is a working day this time. It will be a working day on Sunday this time. Like day after tomorrow is a working day, and that is the reason your test is kept on Monday. I will not cut any part. Whatever I say to every class, even my goal 100 class, I say to you also. And keep on saying that. Okay, bachcho, this is all about today's class. God bless you all. Love you all. And yes, we will meet very soon. And this time, as I promised to you, whatever the optics is, syllabus will be completed. You don't know how much it is always worrying. Uh, okay, beta. Thank you, beta. God bless you. God bless you, everyone. Riger. So this time this is nothing. Riger mortis. हाँ, ये allam chutney है. And yes, by seeing that you understand that when the action potential comes here, there is a marginalization of these vesicle membranes they are biomembranes and they can fuse. All membranes are same. And here we have already that how the thick filaments and this structure LMM, HMM I just said, and I thought that I have to start from the beginning. Anyways, this is our classwork, and classwork everyone should scan it. God bless you, bachcho. Same to you.